Hello everyone, welcome to another video tutorial. So in this video lecture we will discuss about another type of chromatography which is called as gas chromatography. So let's start with the introductive part. So before moving to the gas chromatography, let's understand some basic facts about chromatography. So basically the chromatography is defined as an important biophysical technique, and this is the very important technique used for the separation, identification and isolation of different types of mixture. So basically the chromatography enables the separation, identification and purification of the components from a sample mixture, and this is made up of two words which are named as chroma and graphene. So the word chroma and graphene are derived from Greek words, and the word chroma means color and the word graphene means to write. So at first, the Russian botanist named as Michael Svet coined the term chromatography in 1906, and Svet used this technique for the separation of different plant pigments, and that's why it is named as chromatography. So after some time, the first analytical use for the chromatography was used or described by two scientists named as James and Martin in 1952, and they used a type of chromatography named as gas chromatography for the analysis of fatty acid mixtures. So basically, a chromatography is an important method used for separation of different types of components from a mixture or a sample mixture which have different components. So the chromatography is of different types which are named as column chromatography, paper chromatography, adsorption chromatography, gas chromatography, affinity chromatography, thin layer chromatography and many others. So in this video lecture, we will discuss about gas chromatography in detail. So let's start with the introduction about the gas chromatography. So basically, it is a type of chromatography technique which is used basically in analytical chemistry to separate and to identify different types of components in a sample mixture. Now the gas chromatography is used for separating different chemical components from the sample mixture and then detect them to ascertain their presence or absence in the particular mixture or the particular chemical mixture. So the gas chromatography is quite different from other forms of chromatography because in that condition the mobile phase is the gas and the components are separated as vapor. So this method is used to separate the components or the compounds that can be vaporized without decomposition. Now let's understand about the characteristic features about the gas chromatography. So basically, the gas chromatography is a type of chromatography and it is quite different from other methods. We know that all the chromatography methods include two bases which are named as stationary phase and the another one is named as mobile phase. So the stationary phase and the mobile phase are the basis for all the chromatographic methods, and in the case of gas chromatography the mobile phase is in the gaseous form and here all the components are separated as vapors, and that's why this method is only used to detect some smaller molecular weight having components and which are volatile components, and these compounds having small molecular weight and they do not decomposition in the process of vaporization. So basically it is also called as vapor phase chromatography and gas liquid partition chromatography because it include the gaseous phase or the gaseous matter in the mobile phase of the method. Now there are two types of gas chromatography. So depending on the stationary phase used for the analytical technique, this method is categorized into two types which are named as gas liquid chromatography and the another one is named as gas solid chromatography. So basically, when the stationary phase include liquid substance, it is named as gas liquid chromatography, and when it include a solid matter, it is named as gas solid chromatography. And among both of these, the GLC or the gas liquid chromatography is most widely used method for the chromatography. So before moving to the procedure of the gas chromatography, let's understand about the principle and what is the principle on which the method works. So basically all the chromatic methods work in the similar principle. All the chromatography methods have two phases. The first one is stationary phase and the another one is mobile phase, which have the ability of movement. So in the condition of gas chromatography, the mobile phase will be always gas, but the stationary phase can be solid or liquid. So when the GLC or the in the condition of gas liquid chromatography, where the stationary phase is liquid, the mobile phase like helium or argon or krypton are used because these gases are inert in nature. They cannot react with anyone, and helium is most likely used for this method of gas chromatography because of its low molecular weight. And the stationary phase having high boiling point liquid adsorbed onto the solid. So like other chromatography methods, here the mobile phase, which is the gas, is a chemically inert gas like helium, krypton, argon and many others. And these gases carry the analyte through the heated column and they separate its individual compounds. So in that condition, the compounds which have the greater affinity for the stationary phase, they have longer retention time, and those compounds which do not have the greater affinity for the stationary phase have not longer retention time. So now, before moving to the procedure of the working mechanism of the gas chromatography, let's understand about the instruments and the equipments used for this method. So among both of the types, the GLC or the gas liquid chromatography is the most popular method and widely used method for the chromatography. And this method consists of an injection part, a column, a oven and heater, a gas flow control equipment and a detector. So here a injection part used to inject the sample mixture into the column, because the analyte which is need to be separated is used in a very small quantity and it is injected into the machine with the help of rubber septum at the injection port with the use of a small syringe. Now the second instrument is column. So in this method the columns are used and the sample mixture is injected into these columns. Now the third equipment is called as oven. So basically this oven is used to control the thermostatic temperature of the columns between 50 to 250 degrees Celsius, and this oven doesn't allow the heat transfer from or into the column, and that's why it maintains the temperature of the column. Now the heater and a gas flow control equipment are also used to control the temperature of the column. And the final instrument used for the gas chromatography is a detector or a recorder. So basically detector is used to detect the separated components, and the flame ionization detector is the most commonly used detector in the gas chromatography. So here this type of detector use organic molecules which are burned by flame to get ions and electrons. So these are the instruments used for the gas chromatography. Now let's discuss about the steps of gas chromatography. So basically the gas chromatography mechanism includes the sample injection and vaporization, which is followed by the separation of these components in the column and then detecting the results and recording the results. Basically, whole procedure completes in a automated machine where the sample is injected by a small syringe, and this is injected by the injecting part into the columns, and then it is vaporized, because we know and we discussed in our earlier slides that in the process of gas chromatography the mobile phase is gaseous matter, and it includes vaporization of the mixture. Here, according to the affinity, different components of the mixture are separated in the column and then they are detected by the detector and the results are recorded, and after that the results are analyzed and identified the components found by the mixture. So now let's discuss about the applications of gas chromatography. So basically this method is widely used for the analysis of different air pollutants like sulfur dioxide, carbon dioxide and many other air pollutants, and it is also used for the analysis of different types of essential oils which are extracted from different plants like lavender oil, rose oil and many other oils which are extracted from different plants and flowers. The another application of the chromatography is used to measuring the toxic substances and also measuring the toxicity in the soil and water. That's why this method is widely used by the scientists, environmentalists, researchers to measure the toxic substances in the soil and also measure the pollution in the soil as well as in water. The another application of gas chromatography includes it is used extensively in the field of forensic science. So basically it is used as a solid drug dose identification and also in quantification or paint chip analysis, different toxicology cases and some crime scenes, and in different other functions the gas chromatography is used. The another application of the gas chromatography used to calculate the content of the chemical product in different labs. So the gas chromatography is very accurate if it is used properly, and there are very low chances of wrong results with this chromatography method, and that's why it is widely used in the industrial field as well as in laboratories. Besides this, there are several advantages like it is favored non-polar molecules also, and it is typically used in different applications where small and volatile molecules are detected, but it is not applicable for large molecular weighted molecules. So beside all the advantages, there are some limitations also for this method, that the samples are also required to be salt free. So these are the limitations for this method, that it can analyze only those compounds which have less molecular weight, because we know that the method includes vaporization of the mixture. So for vaporization it have or it should let less molecular weight. Now the another limitation include the compound should have vapor pressure greater than 0, and a very minute or small amount of substance can be measured by this method. So this method cannot work in the large amount of substance which to be separated. And the another limitation includes that the samples which are to be separated should be always salted free, and they should not contain any ions. So these are the limitations of this method. So in this video lecture we have discussed about the gas chromatography, its procedure, its applications and limitations of gas chromatography.